I was always very attracted to art, to the sciences as well, but it wasn't really at the forefront of my mind until I got diagnosed with cancer at the age of 18. And I spent about 2 years in and out of hospitals, at which time I started thinking of how I could give back to the medical community. And that's when I decided to pursue a scientific career and find a path in which to hopefully influence and make the life of other patients better. I got into the University of Florida and was surrounded by this wonderful team of early pioneers in the gene therapy space. The simplicity of gene therapy is what the name implies. You're creating a therapy with a gene. So if you think of people that are born with genetic disorders, they have a faulty gene. And so what gene therapy really is to try to correct the gene itself, either by correcting the gene or by delivering a healthy copy of the gene. You can deliver these genes with what are called non-viral modalities, and there's ways to deliver these genes with viruses. Some of the most exciting things happening in gene therapy right now are happening with viruses. Viruses have evolved over billions of years to attach to a cell, get into the cell, and then deposit their DNA. And if it's harmful, it will hijack that cell, and this is what causes disease. We use a virus that's completely non-pathogenic, meaning there is no known human disease associated with it. And we're able to take this virus shell and clone, for example, a copy of the healthy gene that a patient's missing. When the virus gets into the nucleus of a cell, it's depositing a healthy copy of the gene that's missing. And that results in a therapy. So one of the areas that we're going to be working at Sanofi is called capsid evolution, and that's basically the evolution of these viral shells. This virus actually forms this 3D shape, not unlike a soccer ball, but it's formed of equilateral triangles, 20 of them, to form this shell. Inside the shell, we have our DNA, but capsid evolution is basically trying to evolve this shell. And what we can do with capsid evolution is change these proteins on the surface with millions of permutations, and then select the permutations that do give this capsid now the ability to enter cells that we weren't able to enter before. Through capsid evolution, we can generate new capsids that can enter the muscle cell with 10 times more efficiency than current ones. I strongly believe that the area where gene therapy is going to have the most and the biggest immediate impact is in the rare disease space. Each rare disease has its nuance. We might change the shell of the viral vector to, instead of targeting the liver, we now change that shell for a shell that is better suited to enter neurons to treat diseases like ALS. Gene therapy is a new type of medicine. It's usually a one-time therapy, so this is disruptive. It's having a huge impact in diseases that traditional medicines weren't able to tackle. I wanted to try to make a difference for patients because I had lived it. I spent 2 years in a hospital, and I knew, that sometimes you just need that hope that there's something in the horizon that might get rid of your disease. And, when I was sick, I was very grateful for the advances in the field that I was treated, and I felt like I wanted to give back.